All right. So welcome to Bioinformatics 101 seminar series. My name is Amy Olex. I am a bioinformatics specialist here at the Wright Center for Clinical and Translational Research here at VCU. Apologies for the slight late start. I will try to end on time for everyone's schedules. So before we get into the content for the lecture, I just wanted to go over a couple of seminar logistics. I'm doing things slightly differently than last year, if anyone came last year. All of the presentation content, the syllabus, as well as the quizzes that I'll be passing out after the lecture are all on a Google Drive that I've tried to share with everyone who has registered. If you are attending and have not registered and would like access to that, please email me. Communication will be mostly through email. I have a mailing list set up with everyone who's been registered. So again, if you're not registered and want to receive communications, please email me and I'll add you to that list. For those of you going for the certificate of completion, you'll need to come to 6 out of the 8 lectures and either attend in person or online. Then after each lecture, there will be a quiz sent out. You'll have a week to complete the quiz. And if you score a cumulative score of 42 or greater on all of the quizzes that you take and are able to attend 6 of the 8 lectures, then I'll be issuing certificates of completion. There'll be digital PDFs that I'll send out after the last week of the seminar. So attendance for the certificate, I make it mandatory that you either come in person or are online so that you are here and are able to ask questions. If anyone online needs to ask a question, I am monitoring the chat, so I will do my best to answer any questions online because we can kind of hear some people, but it's not real good audio quality on my end. Also, I'll be sending out surveys after each session. These are for quality improvement and to get your feedback on how the session went, and if there's any topics that you would like discussed possibly in future workshops or seminars that the CCTR hosts. So this lecture is going to be an introduction to bioinformatics starting from my perspective. I was introduced to bioinformatics in 2005. When I was in my senior year of undergraduate. I was a computer science major, looking for something else to do with my computer skills other than sit in a cubicle and be a code monkey for some company. So I was always interested in biology, and so I discovered bioinformatics, took the introductory course, and just fell in love with the field. And back then, my impression of bioinformatics was computer science intersecting with biology, where you use computational methods and techniques to store, analyze, and organize biological data. Since then, and through my experience working in bioinformatics since 2005, I've kind of seen an evolution of the field of bioinformatics occur. The first phase I call classical bioinformatics, and this primarily occurs before the first human genome was sequenced, before high throughput technologies such as microarrays and high throughput sequencing came into the scene, and mostly focused on characterizing molecules and sequence analysis and identifying mechanism at the molecular level. So there's actually little use of computers other than visualizing data and storing some data. And looking back even further, bioinformatics was originally coined in the 1970s by Pauline Hodgway, and she described bioinformatics as the study of informatics processes in biotic systems. So originally, bioinformatics really had nothing to do with computers. It was about studying how the human body or biology in general stores and processes information. Now, with technology advancements and the ability to gather more and more biological data, you need computers to store and process that data so that you're able to analyze and interpret it. So bioinformatics has started to evolve to include the development of computational methods specifically for analyzing biological and molecular data. The data that bioinformatics mainly focuses on includes the genome and genomics, gene expression with transcriptomics, proteomics, all the way from your DNA up to systems level with signaling pathways and such. And so all of these different data types are the focus of bioinformatics. And with all of these different data types and the advancements in technology of how we can capture and process this biological information, many other fields have come into bioinformatics to make it a highly interdisciplinary research field. So now we have computer scientists, biologists, statisticians, mathematics, physics, information technology has come into the works because you need that information infrastructure to be able to store all of this data. And now we're seeing the medical and clinical data also coming into bioinformatics. And this is actually shown every year. This is the 4th year I've offered this seminar series. This is the backgrounds that you entered when you registered for the course. And every year I see this wide range of backgrounds where we have people with physics and mathematics backgrounds, medical and clinical. Generally, for the past 4 years, the biology, molecular biology background, a lot of people have that, which makes sense as this field of bioinformatics kind of emerged from the molecular biology. So in my view, and when you ask different people, you might get different responses, but in my view, bioinformaticians fall into 3 primary roles. You have the researcher in bioinformatics who are focused on theoretical and algorithm developments and advances in bioinformatics methods. So those are the actual algorithms that can do sequence alignment, that can process a protein structure and do predictions for protein structure and folding and that kind of thing. Then you have a developer who focuses more on infrastructure development. So not the design and development of the algorithms, but maybe the most efficient implementation of an algorithm. Also developing tools and applications that researchers can use to process and analyze and store their information, and building computational infrastructure to store all of this data. Then lastly, you have the user. The user is focused a lot on the application of bioinformatics tools and methods to analyze their data, to look at their biological problem that they're interested in. And so one bioinformatician can be all of these or mostly one of these, and it really depends on your background, where you came from, what your research goals are, and such, to which realm you primarily fall in. And here, again, if you ask another bioinformatician, again, they came from a different background, have a different experience, they may see bioinformatics in a slightly different way than I do, but at the core, bioinformatics is still about understanding how biological systems store and process information, and how we can use that knowledge to further clinical care and advances and that kind of thing. So I pulled up a few differing definitions of bioinformatics just to show the diversity of how different people can define bioinformatics in a slightly different way. The first is focused on mathematical and statistical computing methods to solve biological problems using the DNA and amino acid sequence. So it's rather focused on using DNA and amino acid sequences, such as solving protein folding problem. Can you take a protein sequence and predict what shape it's going to fold up into? The second is where bioinformatics is the field of science in which biology, computer science, and information technology merge to form a single discipline. This is more of where I came into bioinformatics and viewed it as a merging of computer science and biology. And the goal is to discover new biological insights, as well as to define the unifying principles in biology. Lastly, the NIH defines bioinformatics as research, development, or application of computational tools and approaches for expanding the use of biological, medical, behavioral, or health data. So we go from analyzing and trying to figure out how biology stores and processes information up to now we have knowledge of how it does that, how can we expand the use of that knowledge into the medical and clinical realm to affect patient care. So the second phase of the evolution of bioinformatics I call post-genomics. This is after high-throughput technologies has come on the scene, such as gene expression microarrays, high-throughput sequencing, and now we have so much information that bioinformatics has started to focus on being able to extract information out of that data that we've collected, analyzing thousands of sequences simultaneously, collecting thousands and thousands of sequences for populations to characterize diseases among certain populations, and also integrating many different data types, genomics, proteomics, lipidomics, metabolomics, all these other different data types. So this phase could also be called the multi-omics generation of bioinformatics. And now we're trying to expand these data types and also include clinical data types as well. So an example of this phase of evolution I found is the 2014 ALS Ice Bucket Challenge, which many of us may remember, went viral on the internet. So this challenge was meant to raise money for ALS, which is a devastating neurodegenerative disease that can lead to paralysis or death. There are very few research directions or targets for therapeutic targets that researchers had at the time. And so they raised $220 million that helped create an international network of researchers to look at ALS and try to identify new targets. And out of this actually came a new gene called NEC1, where variants in NEC1 were found to be strongly associated with ALS. And further research with NEC1 shows that the NEC1 protein actually interacts with a lot of other known ALS-associated proteins. So this challenge actually provided a new target for developing ALS therapies. So how did they do this? How did they find this gene? Well, they actually had to sequence 14,000 people with ALS and process and analyze that sequencing data to find this one NEC1 variant. So this kind of brings us to bioinformatics in the era of big data. So 14,000 genome sequences. How much data is that they had to store, organize, process, and sift through to find this one variant in NEC1? Well, there were over 14,000 genome sequences. We if we assume each person takes up 100 gigabytes per genome, that's about 1,400 terabytes of data, which is equivalent to over 43,000 32 gigabyte iPhones. That's a lot of data. So we need better storage and better infrastructure in order to organize and integrate all of this data so that we can pull out information and knowledge in order to identify the NEC1 gene or other targets. Another example of this big data is the Genomic Data Commons, which the GDC's mission is to provide the cancer research community with a unified data repository that enables data sharing across cancer genomic studies in support of precision medicine. So one issue that we have with big data is we have all of these labs and research studies that collect the data using a certain protocol, using certain bioinformatics methods, and we have all these silos of projects where they're able to collect some samples. Zoom recording, okay. And they're not seeing me either. So, can you see me now? Somehow mute and stop video got.
It looks like it got stopped and it's still trying to record.
I think so.
I was trying to save the recording.
Oh, what?
Just
That's not, okay. All right. It should be started. It's started. It's recording. My apologies. I don't know what happened. I think they can see me now. You guys can see me now? When did that happen? Oh boy.
Is your family okay?
Can you guys still hear me? Okay. All right. My apologies. I'm very sorry. All right. So, all right. Informatic. My apologies to everyone here too. That's never happened before where it decided to just mute and stop video all by itself. So, all right, so informatics. All areas of informatics have really a core process where you acquire and store and organize data. Then you transform that data into information, and that information gets translated into knowledge, which is then actionable. For example, advertising survey results for a business. So those survey results are your data, and then you analyze those and identify that the newspaper is the lowest reported ad. That's your information that you gain from that data. Then you interpret and learn from that information. This is mostly manual, using your own intuition. And then you interpret and figure out that, well, maybe few people read my ad in the newspaper, so what are we going to do about that? Well, let's stop buying ads in the newspaper. It's not really productive, so that's your decision and your action, which then changes the data. In a bioinformatics terms, your data would be your gene expression results, so just the expression data. Then you analyze and visualize that, maybe cluster it, and you identify genes that are differentially expressed. Maybe some are upregulated, some are downregulated. You have clusters of genes. Then you use your knowledge of the biological system to interpret those clusters and those genes that are differentially expressed to learn about your biological system, of which then you can hypothesize what's going on and design new experiments and to create new data, which changes the data, and the cycle starts over again. So bioinformatics in this stage is really data-driven bioinformatics. This word cloud was generated from 1,000 abstracts in the Bioinformatics Journal in the year 2014, and the top 100 words were put into a word cloud, where the size of the word is means the word was more frequent. And we can see the most frequent word was data, and then we have the types of data: gene data, protein data, expression data, sequencing data. So I really like this word cloud because it kind of summarizes what bioinformatics is, that it's focused on data and the type of data that it's focused on. So this is just a rather incomplete list of bioinformatics research at VCU, and the point I want to make is just the interdisciplinary nature of bioinformatics. So we have Dr. Aradz and Dr. Kurgan, who are actually in the computer science department, who focus on the theoretical aspects of bioinformatics algorithms. Then Dr. Dosmarov is in the Department of Biostatistics. We have Dr. Miles from the Center for Structural and Biological Complexity, as well as Dr. Fetwace and Dr. Buck are also from that center. Dr. Webb is from the Department of Psychiatry. Dr. Morgan is from the School of Dentistry, and I've worked collaboratively with him on a project studying the human papillomavirus. And Dr. Harrell is in the Department of Pathology, which we've been studying breast cancer PDX models. So this is just to give an example that bioinformatics plays a role in many different disciplines, and it's just very highly interdisciplinary. So it's a fun field. I like working in bioinformatics. This slide is just to go over 3 fields that very commonly get confused and interchanged. And to me, they're 3 different fields. So there's computational biology and bioinformatics. When I first started in bioinformatics, I kind of thought these were the same thing. They were synonyms for each other. But since I've come to learn that computational biology is really more focused on predictive modeling of biological systems. So you build a mathematical model of a biological system where you can simulate stimuli to that system and predict how the system is going to react. Whereas bioinformatics is just taking the data that you've collected from your biological system and analyzing it and visualizing it and help you interpret that data. So computational biology overlaps a lot with bioinformatics for sure. Computational biology can use the data that bioinformatics can extract from the actual system, and then computational biology goes further and does the predictive modeling of that system. Biostatistics is more focused on statistical inference, identifying your sample size and that kind of thing. So biostatistics is more closely related to statistics rather than computer science or informatics. But again, they all deal with the same biological data, so they do overlap a lot. Okay, I'll try to speed up a little bit here. So the last evolution of bioinformatics is after the $1,000 genome, and this is the era of precision medicine. It's focused on an individual, and it combines personalized genomic data and clinical information to aid physicians in clinical decision making. So the $1,000 genome, originally, when we first started sequencing, it cost $3 billion to sequence the first human genome, and over 10 years, I think. Since then, and with the advent of high throughput sequencing, the cost of sequencing has dramatically dropped, and I think at this point we're down to about $1,000 per genome. With the drop in price, of course, the number of experiments that are run goes up, so lower cost plus higher throughput equals lots and lots and lots of data. So precision medicine, it focuses on an individual's information. It focuses on the prevention, diagnosis, and treatment of disease based on a single individual's genomic, molecular, and environmental information. So this precision medicine targets patients who will benefit the most from preventative and therapeutic interventions. You want to use that person's biology, that person's genomic information to figure out, will this drug actually help them, or will it hurt them, or will it not do anything? So precision medicine is becoming data-driven. You need genomics and proteomics and metabolomics and imaging and biometrics and environmental factors for your patient. And you need to be able to merge all of these different data types in such a form that a physician can use them to make clinical decisions on a patient's care. Also, some people see that precision medicine is moving from reactive to preventative. So by reactive, I mean you're sick, you come in, you found you have cancer, they go and sequence the cancer, figure out if any particular targeted therapeutic drug may benefit your cancer and give you therapies based off of that. You're reacting to the disease. Preventative is where you would sequence your genome at birth, and there are projects out there that do this. I think one of them is called BabySeq, where you're born and the first thing they do is sequence your genome and figure out, do you have any of the variants that are associated with higher probability for breast cancer or other diseases like that. And then you can do preventative things with your lifestyle or medications or whatever to try and prevent those diseases from occurring. So last year I went to a boot camp on precision health from Olex to data science. It was a very intense week, and I learned a lot from it. And I learned that there are a lot of different facets to precision medicine. You have the clinical and patient care, which is focused on the patient and the clinician, and how the clinician can best use the information to make decisions about how to treat their patients. Then you have informatics infrastructure, where you need computational infrastructure to store and organize and process and secure patient data. Then you have the translational aspect, where you have bench discoveries that you're making in the lab, but they're not directly applicable to application in the clinic. You have to go through this translational process to be able to take those bench results and help the physicians understand how those results may impact their patient. Educational, there's a lot of educational aspects to precision medicine. You educate the clinicians on informatics and the type of data that informatics generates. This is genomic data, sequencing data, what variants, how to interpret those. And then there's the patient. You tell a patient you want to sequence their genome. What does that mean? What are you doing? What is the data? You know, where's the data going to go? Who's going to see the data? If the data gets out public, what could happen? That kind of thing. And then the bioinformatician. When you're handling patient data, there are a lot of rules and regulations that are in place to protect the patient's privacy. HIPAA, of course. The bioinformatician, this is not plant data, it's not cell types that you're dealing with, it's actual patient data. So the bioinformatician needs to know how to properly store that data, how to process it, who they can let see it and who can't see it. There's a lot of regulations like that. And that leads into ethical considerations, where a person's genome, you sequence their genome, that's protected health information. That's who they are. You can't really de-identify their genome. So there are a lot of different rules and regulations that come into play when you're dealing with patient data. And everyone across the board, researchers, bioinformaticians, clinicians, patients, all need to know how that data is going to be stored and handled. So where does bioinformatics fit into precision medicine? Just about everywhere, really. So the key data and the cornerstone data in precision medicine is omics data. The genome, a person's DNA, that's the data that bioinformatics is focused on analyzing, and that is the data that you use for precision medicine to figure out how an individual is going to react to something. So in reactive precision medicine, you characterize different diseases, and there are personalized targeted cancer therapies. So Keytruda is one company that offers a targeted therapy, where if you have a certain variant in a certain gene, then you can use their therapy because it targets that specific variant. Preventative and predictive is you're trying to characterize your genome and identify things so that you can change your lifestyle or behavior before symptoms show up. For example, personalized nutrition. The company Veritas Genetics actually sequences your genome and looks for specific variants in your genome that may cause you to metabolize food differently. For example, caffeine. If you metabolize caffeine at a fast rate, you can drink all the coffee you want and you might not know the difference. But if you don't metabolize caffeine that well, or you're a poor metabolizer, caffeine may cause complications. I'm one of them. I get jittery and shaky when I have just like one cup of coffee. It's unworkable. So I avoid caffeine at all costs. Preventative or predictive is you try to look for things in your genome and identify areas of your life that maybe you can change your behavior, change your diet to change your outcome. So this is just an example at the workshop that was presented by Dr. Valentine, using bioinformatics to detect transplant rejection. Early detection of heart transplant rejection is vital so that you can know if the patient is rejecting or accepting the heart, because you need to take action quickly. Normally, the rejection would be identified using regular biopsies, where they just take a little bit of the heart and process it and see if it's doing well or not doing well. But Dr. Valentine said one of her patients was like, you give me a new heart and then you take it away piece by piece. So they came up with a method to identify transplant rejection non-invasively. So Dr. Valentine said, if you think about it this way, an organ transplant is essentially a genome transplant. You're having someone else's DNA with all of their variants being put into your body. Is your body going to accept it or not? And so this is one reason why they try to give a woman a woman's heart and men a men's heart, because you have that XY chromosome difference, which can, I guess, cause major complications. So what they did is they monitored the rejection looking at cell-free DNA. So cell-free DNA comes from apoptotic or necrotic cells that are dying, and it circulates in the plasma. So what they have is they have the patient, and they just take a sample of the plasma and they identify the cell-free DNA that's in the plasma. They sequence it, and then they can identify whether it came from the person or the actual heart, the donor. And they do this by first genotyping the patient's DNA and then genotyping the donor's DNA and identifying key SNPs and places of variation in the genome that are different between the two individuals. And then they can pull out these DNA fragments from the serum and the plasma and sequence them and look for these SNPs precisely and be able to identify how much donor DNA is floating around in the bloodstream. And they found that when they're monitoring the donor DNA, they notice an increase in donor DNA when the heart's going through rejection a lot sooner than what the biopsies show. So they're able to act sooner and faster and recognize the sign of rejection. So we just went over the bioinformatics methods. You have to sequence and genotype the donor and the patient, and to be able to calculate the percent of cell-free DNA from the donor. Another example is in the field of pharmacogenetics, which is the study of how the genome affects an individual's reaction to drugs. So there are actually over 190 drugs that are FDA approved with genetic labeling, saying that if a person has this variant in this gene, then they may react poorly or they may overreact to a given drug. And so CPIC, which is the Clinical Pharmacogenetics Implementation Consortium, has written down easily readable guidelines for 33 out of these 190 drugs. And they've written it in a way that helps physicians interpret the information to optimize giving the drugs to patients and figuring out if a patient should take a drug or not. For example, clopidogrel is used to treat blood clotting, so it has antiplatelet activity, but in order for it to serve in that manner, it has to be metabolized into another form by your body. And so patients that have two loss-of-function alleles in the gene CYP2C19 don't metabolize the drug well. So if the drug is not metabolized, then it doesn't get transformed into its active form, which means it's not going to do anything for the patient. So what CPIC has done is it's taken the FDA drug label and converted it into a nice handy little flow chart here that says if your patient has this genotype or has a particular SNP, say it goes under the which is an ultra metabolizer. That means they're going to metabolize that drug really fast and convert it into the active form. That's great, that's what we want. Give them a regular dosage. Whereas if they're a poor metabolizer, they won't be metabolizing that drug very fast, so it's not going to be converted to its active form. So you may want to consider a different class of drugs. And there are these flowcharts for 33 of the drugs, and some of them even say, you know, if you're a poor metabolizer or an ultra metabolizer, this is dangerous to the patient. They could die. They could, if they metabolize the drug too fast, they're going to get a drug overdose kind of thing. So this helps patients or clinicians actually easily look at, okay, the patient has these variants, and this is what it means clinically, and this is how I should adjust my dosing schedule. The University of Florida has actually implemented these CPEC guidelines in their clinics, where if a patient is, say, prescribed clopidogrel, they will get a message box up asking, have you done genotyping of the patient? Yes or no? Do you want to do genotyping of the patient before you prescribe this medication? So they actually have to take an action, and they're aware of these implications. So from bench to bedside, bioinformatics fits in where in the sequencing the gene, identifying the variants, looking at the impacts of the protein sequence. If you have a variant, how does that affect the protein sequence? And how does the change in the protein sequence affect the structure and function of the actual protein? And then how does the altered structure and function of the protein affect, say, reaction to drug or signaling pathways? And so bioinformatics deals with all of these areas. And the question is, what are the clinical implications of this bench discovery? And that's the area of translational bioinformatics, which is translational bioinformatics focuses on taking these bench discoveries and storing them, analyzing them, and visualizing them in a way so that they are useful for clinicians to utilize in their patient care and decision making. So some of the added challenges that translational bioinformatics includes is looking at patient privacy, informed consent, security of the data standards, and a host of other challenges that come when you're dealing with data in a medical environment. But the focus is primarily on that translation of bench discoveries into the clinic so that those discoveries can be actionable. Okay, just to recap, almost done. So bioinformatics, as I see it, is evolving, and it's still evolving. We started at looking at how biological systems store and process information. And then with the advent of high-throughput technology and advancements in computation, it now includes computational methods and the infrastructure to store and process that biological information that's collected. And now in the era of precision medicine, we are focused on utilizing the knowledge that we've gained from those discoveries in clinical decision support. So, but ultimately bioinformatics is still, and I think always will be, rooted in understanding how biological systems store and process information. So it's highly interdisciplinary, and it is clinically relevant at this stage because we can sequence a genome quickly and cheaply. I think bioinformatics is a cornerstone of precision medicine. I'm not sure you can do precision medicine without implementing or using data from some type of bioinformatics analysis or discovery that was made through bioinformatics analysis. And it's really focused on transforming the data into knowledge. And in precision medicine, we go one step further where we take that knowledge and transform it into actions and clinical decisions to affect patient care. So, and also the type of data that separates bioinformatics from other informatics is that storage and processing of biological information. And so this is my last slide for the presentation. This is an endless cycle. Bioinformatics transforms data into knowledge, which we make to use decisions. Our decisions then change the data, and the cycle continues. This is just an overview of the seminar series that we'll have this week. I'm still working on the last lecture, but there will be a lecture there. And if any of the information that you've learned has helped in your research throughout the entire seminar series, please consider citing the CTSA grant. It's what funds my position in the seminar series. And here's just some resources that you can use if you need help with bioinformatics. Thank you very much, and I will take any questions. Yes.
You know, like when you're talking about precision medicine and, you know, all of that, and that's great, but, you know, where do the insurance companies come in on funding for a lot of this genomic information to make clinical decisions? I know that's not necessarily your wheelhouse, but I think that's a consideration in how this information is more publicly used.
Yeah, so insurance companies are a bit of a sticky point because there's a lot of I guess fear that if you get sequenced from birth and the insurance company knows, say, that you're going to develop ALS, you know, when you get older, are they going to insure you from birth or refuse you insurance as an infant? So, yeah, insurance companies, I'm not sure how much they fund the research, but there is a lot of, I guess, patient privacy concerns and such. If your genomic data is made public, can the insurance companies use that to deny you coverage?
Or even like in the drug,
I think what Shirley is that if you use precision medicine in treatment, then is it funded by insurance? And it is actually, so there are many drugs that are already funded.
I mean, I think that's changing, but I think it depends on your insurance company. You know, I don't know what Medicare and Medicaid does, and how equitable that is across. You know,
it's getting there because they actually realize that it's cheaper for them to pay for the better drugs, because on the long term, you know, if it cures cancer, then you don't have to pay for another year for the treatment. So.
Yeah,
but there are. I mean, you guys probably know this better, but there are.
So there's definitely some variation in terms of what will be covered for well-validated, actionable mutations. Testing is essentially universally covered by insurance, covered by Medicare, and private insurance companies tend to follow suit. I think where things get a little bit more difficult is kind of where you're going off the beaten path, and you know, if you order a sequence, you know, a panel of several, you know, typically mutated genes, looking more for not kind of standard of care treatments, but more, is there something else that we can try, off-label usage or candidacy for clinical trials? That's where things get a little bit more difficult with getting insurance to necessarily cover. But even in those situations, oftentimes they'll play ball with you.
Like Foundation One or Oncotype, those are standard of care, FDA approved things. So insurance companies won't be able to refuse. I mean, they can refuse, but you know, you can have a strong case. But if something is not FDA approved and the patient's insurance is sort of soft and things like that, then yeah, you can certainly run into trouble.
All right. Well, thank you. Next week, we're actually going to do the seminar in the adjoining room because there's some training going on in this room, but then the rest of the time, the seminars will be in this room. If you didn't sign the sign-in sheet, it's up here before you leave. Otherwise, thank you.